the title sounds very technological, but what I actually want to do is to walk you, and this will be a quick ride, through our journey of developing mRNA as a technology to be usable preventively and therapeutically in humans. And this journey actually started already in the 1990s when we BioNTech founders were young physicians treating cancer patients and realized that how high the medical need is, and that there is not much what we can do for our cancer patients. And we understood that we need different modalities to treat cancer, which is a very complex disease because it's basically unique in each and every patient. And we were immunologists and therefore fascinated by the armamentarium the immune system has to combat all types of diseases. And so it became, back then in the 1990s, our declared vision to develop a novel type of cancer therapies based on understanding exactly what the features are which distinguish cancer from normal cells in order to have a target, an antigen, to direct the immune system against, also to develop personalized approaches, because, you know, cancer in every patient is unique, and it is an interesting vision to think about truly personalizing cancer therapies. And the third interest was to develop more potent immune therapy platforms, including vaccines. When we were looking for different methods of formats for vaccines, and back in the 1990s, people were trying DNA, protein, peptides, so different chemistries, we came across mRNA, and comparing these technologies, it became very clear to us very early on that this was a technology which had a couple of advantages which could make it to a breakthrough in immune therapies. mRNA, messenger RNA is actually not a synthetic artificial molecule. It is the most primal information technology which has been created by nature. We have mRNA in each and every cell, and it is a messenger or a courier of information. Our DNA, which is the fixed genetic material, the hardware, so to say, is translated into the molecules of life, which are responsible for all sorts of life processes, proteins, via the intermediate mRNA. And mRNA is, so to say, the template for proteins, is there transiently and then disappears. And mRNA was discovered many years ago, in 1961, by several scientists, and since then many generations of scientists have worked on understanding mRNA, how it works, but also making it amenable for therapeutic and preventive purposes. One of the immune therapy approaches we were interested in was cancer vaccines. Cancer vaccines is a bit different from what we know about vaccination in infectious diseases. In cancer vaccines, you have a situation that the tumor, the cancer is already there. And sometimes the burden of cancer cells is high. You have billions and billions of cells, and you have to build a very strong immune response by the vaccine in order to combat these cancer cells. And there are a couple of requirements which you need for a cancer vaccine. First of all, just to explain to you, what does cancer vaccine mean? It means in principle that you expose the body to the features of against which you want the immune system to attack. And this is in general done in the protein format. So these features are molecularly presented as protein to the immune system so that it understands what to attack. And the requirements for a cancer vaccine to function are that you have to bring this information of the features to special cells of the body, which are called antigen-presenting cells. And these are, so to say, the generals of the immune system, which instruct immune cells what to see as the enemy. The presence of the antigen, the target, in these professional antigen-presenting cells has to lead to the induction of an immune response. And we need very special cells to be induced here. These are called T cells. These are killer cells, which specifically instructed by the generals of the immune system, by the antigen-presenting cells, with high precision, recognize those targets, those antigens which you have presented. That's not sufficient for a vaccine. A vaccine has also to come with what is called adjuvanticity. So with the instruction to not only recognize what is presented as a target, but actually to attack. And this is what we call an innate inflammatory immune signature. And when we were testing mRNA, we were convinced that all these requirements could be addressed and fulfilled by mRNA. The last one, the adjuvanticity, was already there. The mRNA is made out of a 4-letter code, and one of these letters, the so-called uridine, is a danger signal to the immune system. So this was basically already delivered with the mRNA intrinsically. What, however, back in the 1990s was not addressed by mRNA was that we needed the robust presence of the antigen encoded by the mRNA. mRNA was not stable enough. It was at that time not capable of translating with high performance into protein, into the antigen to present, and therefore also did not induce potently the T-cell immune response, the killer cells we needed. And so it was clear for us in the 1990s that these were areas which needed to be addressed by advancements, by discoveries, by inventions in order to make mRNA suitable as a vaccine. And there are, in principle, 3 areas of advancements which were important for us and which we achieved over the last 3 decades in order to make mRNA fit for the purpose of a therapeutic or preventive vaccine. One of these areas was, as I pointed out, the instability of mRNA and its weakness in providing protein translation from the template it delivered. And this again meant that the immune system was not sufficiently stimulated, and the army of killer cells, which we wanted to expand in these patients against the antigen, was also not expanded. And we found out that there are structural elements in the mRNA, and this is shown here. So this is the template from which the wanted poster, so to say, the antigen, the target against which to direct the immune system, is represented and encoded. But there are also these structures here which are responsible for how performant the mRNA is in producing the protein we need. And we found out that by special design, by molecular engineering of these structural elements of mRNA, we can improve the translational efficiency, and also the stability, the half-life of the mRNA, as depicted here, and thereby ensure that we get the amounts of protein we need. And we had to find these designs for these structural elements by high throughput screens, and some of this data is shown here. You can, for example, see here an experiment where we have modified this part, which is called the 3' UTR. This was the 3' UTR which was used back there conventionally, and we found by high throughput screens different designs which, for example, this one were superior in providing the protein for a longer time and at higher amounts. Another area of improvement was the place where to deliver mRNA. In vaccination, location matters. It's not only sufficient to bring the antigen, the wanted poster, so to say, to the antigen-presenting cells I just mentioned, those generals of the immune system. The mRNA also has to go to very special locations where advanced versions of these antigen-presenting cells reside. And these are the lymphoid tissues, which are distributed all over the body, the spleen, for example, but also lymph nodes. And back then, when we started cancer vaccination research, different routes were used for vaccination. Into the skin, so intradermally or subcutaneously were the most frequently used routes, and we assessed how these routes would compare from injecting directly into the lymphatic system. And we did this by injecting into lymph nodes of mice. This is shown here. And this so-called intranodal injection resulted in a much higher number of circulating T cells, these killer cells, which we need to kill the cancer cell, as compared to the conventional ways of doing this. And we asked the question, rather than injecting into lymph nodes, which is an inconvenient way for the clinician, but also for the patient, can we find a way to deliver even body-wide into the entire lymphatic system? And for this, we developed a liposomal technology, so-called RNA lipoplexes. So a liposomal formulation in which the mRNA was packaged was intravenously administered, and due to the very specific charge of these particles, we were able to target lymphoid tissues, for example, the spleen. We observed that we need a very special charge, which is neutral, to get into the spleen, as opposed to where you normally get into lungs, for example, of mice. This formulation, these intravenously, and this is for the biologists among you, which is administered intravenously, not then here in the mouse systems, specifically into resident antigen-presenting cells in lymphoid tissues. It also activated them to be more potent in instructing T lymphocytes. This is shown here. And this led in mouse models to strong immune responses, strong T cells directed against tumors, and in difficult to treat mouse models to beneficial treatment effects. But not only in mouse models, we have this technology also in clinical settings. Hundreds of patients with cancer are treated with this intravenous RNA LPX vaccine technology, which is this is one of the examples of our trials where we have treated melanoma patients with mRNA RNA lipoplexes which encode for 4 different melanoma-specific, so tumor-specific antigens. And when we inject these, we see that directly after injection, after a couple of hours, this is the spleen, metabolic activity and proliferation of immune cells is activated. We see exactly that innate immune stimulation which we need. We see that we can expand the killer cells, the CD8 positive T cells, which first, before starting the treatment, are present. We can expand them to 10% of the circulating killer cells of the patient. And as you can see here, we can, by adding this vaccine to an immune therapy, which does not work in this patient, we can achieve tumor regression and partial responses. So this was the second area, the delivery of mRNA-based vaccines to the right places. And the third area is, as I pointed out earlier, in cancer, we were interested in developing personalized vaccines, so truly personalized, tailor-made for the individual patient, simply because that is the root cause why cancers are so difficult to treat, and many therapeutic modalities result in resistance and refractority to treatment. And to address this uniqueness of cancer, we set up an individualized approach, which starts with collecting the patient's samples and analyzing the distinctive features of the tumor as compared to normal tissues, the so-called mutations or genetic aberrations. What one finds is that in each and every patient, these mutations, these features are different. You don't find 2 patients who have the same set of mutations. And we are using these genetic features in order to design a vaccine which reflects these mutations, these features as targets for the immune system. And this is basically produced on demand for this very patient in an individualized and tailored way, generated as an mRNA vaccine in this lipoplex platform, and then shipped back to the patient for repeated immunization. When we started this, it took for our first patients in 2014, it took us a couple of months for each patient to go through this on-demand manufacturing process. Fast forward a couple of years, 2019, and hundreds of patients treated in clinical trials, each patient and each manufacturing a race against this patient's growing tumor. We have this down to 3 to 5 weeks for each patient. This is actually an example from our first trial, which we conducted with such an individualized vaccine. And I'm only showing this piece of data because this piece shows that the immune responses we generate are long-term. After 400 days and even 4 years, we still detect the immune responses against. Prior to vaccination, the immune system was blind. So even if the patient is not vaccinated for a couple of years here, these are repeated vaccinations, but they stop, the immune responses are still there. And these are 13 patients from the very first study, patients with melanoma, which we have treated with this vaccine. And on this left side here, you see triangles. Each of these triangles for each of these patients are the recurrences this patient has faced. So all these triangles in this patient, for example, multiple recurrences before we start with the vaccine. After we started with the vaccine, you can see that the cumulative frequency of recurrences in this patient population has dropped. And now after 6 years of follow-up, we can see that many of these patients are still lesion-free, which shows the potency of this vaccine. We have created actually an entire toolbox around the mRNA technology. We have different chemistries of mRNA vaccines, the nucleoside modified and the not nucleoside modified one, which we are using for our cancer vaccines, self-amplifying mRNA, which comes with its own amplification mechanism. So after having administered it, the mRNA continues to amplify itself and basically increase its own dose. We have different types of nanoparticles, and how we combine all this depends on the purpose for which we want to use this vaccine. That means that we can address a diversity of purposes. We can, for example, use this platform with a couple of tweaks to tell the immune system not to build an immune response, but to dampen an immune response. In the case of autoimmunity, we have shown in mouse systems that we can, by this approach, cure mice with multiple sclerosis in multiple sclerosis model systems. Or if we want to express an antibody against cancer, we can also use this technology in a different way, where the protein, the antibody directed against cancer, is encoded by the mRNA and produced in the liver of the cells, which becomes sort of the factory for protein production. With all this, I think you can imagine that we felt that we have a very potent technology, and we felt obliged, we felt the obligation when we realized that this fateful last weekend of January 2020, we felt the obligation to try to help with development of a vaccine against this new virus, and we managed to do this within 10 months. In the meantime, as you know, billions of people have been vaccinated, and we also have had variant adaptation cycles for this vaccine. When we had developed this, our COVID-19 vaccine, it was clear for us that it was not just done with developing the vaccine against the wild type virus, but that there would be additional challenges. And one challenge was, which was very clear from the beginning, to stay actionable in the face of a dynamic SARS-CoV-2 pandemic, where the virus would mutate and change. So in parallel to conducting our phase 3 trial for our vaccine in 2020, we started to work with our partners at InstaDeep on an early warning system, which is based on large language models, which in the meantime have become famous, in order to analyze published sequence information of virus mutations and predict based on them variants of concern and also variants of concern which would evade the immunity we would build with our vaccine. And this algorithm was actually capable to predict prior WHO prediction or announcement variants of concern. And when we saw that Omicron would become a variant of concern, we immediately could use this knowledge in order to pivot to development of a variant-adapted vaccine to the first Omicron variant, and in the meantime also for this season to the next one. Another important important challenge was and still is that we all know that the virus will continue to evolve, and what we can see here, this is shown here, is if we compare the epitopes for B-cell, the B-cell epitopes, which means the epitopes for antibodies which are generated by the vaccine, we can see that they have already deteriorated by the mutations. What however stays stable are the epitopes and for T cells, that means the killer cells, which we also use in our cancer vaccine. So that means that rather than having a vaccine which just builds on antibody responses, we also need some component which ensures that we strengthen T cell responses. And based on this, we are developing a so-called T cell string against multiple, which represents multiple antigens of SARS-CoV-2, which will become a component of current SARS-CoV of our current spike-based SARS-CoV-2 vaccine. Another challenge was the supply challenge. When we started in 2020, our vaccine COVID-19 vaccine project, which we called Lightspeed, because we knew we have to be fast. We were basically a company which was prepared for low volume production for small cancer patient populations. We were able to produce 10,000 doses of vaccine at that time. In 2020, it became clear we had to increase our capacities, and we increased to 25 million doses. And later in 2021, where it became clear that the burden of delivering COVID-19 vaccine for the entire population would be on the shoulders of a small number of companies, we turned every stone in order to scale up to 3 billion together with our partner Pfizer, and this was a 300K upscaling, and you can imagine also this was needed a lot of innovation and was also a scientific challenge. And the third challenge, just to add to that, what helped us with upscaling was the fact that we had a very potent vaccine. That means small amounts, 30 micrograms, were already sufficient per dose, which translated to the fact that this small system here, this small production batch holds 8 million doses, which tells us that the capacity which we needed was not so enormous as we know it, for example, for antibodies or recombinant proteins. So the third challenge, the equality challenge, we were very naive back in those times. We thought that, okay, it's our job to deliver a vaccine which works, which is efficacious and safe, and some entity will take care of ensuring that this is then equally distributed, because we were just humble cancer vaccine developers, right? We did not know much about global health frameworks and practices, but we learned fast that global ethical and geopolitical societal frameworks are simply not in place for equality of health. And the first reaction was that we ensured that in 2021, 40% of our vaccine production went into low and middle income countries at cost. But that is obviously not a sustainable model to contribute to equality of health. A much sustainable model is to help those regions to become autonomous, and therefore we decided that we would. Does this work? Can I activate this? I'm not sure, no. I have actually a video, but that does, I think, not work. I can go to this. And this motivated us to come up with an idea which first was science fiction-y, but the entire team worked on that, so it became reality, namely container-based, modular, small production facilities, miniaturized, semi-automatized, with cloud-driven production programs, which we call BioNTech, which are assembled in our facilities in Germany and pre-tested and sort of informally certified by us, and which can then transported into those regions in which we want to help to build production, local autonomous production ecosystems. And these containers, which are basically the normal shipping containers, we need 4 of them in order to cover the entire mRNA production, need certainly an ecosystem, so that what we need from our partners in those regions are utilities, access to talent, regulatory frameworks, operation permits, fill and finish capacities, logistics and supplies. But we are committed to do our part to that. And this started as a vision, as a dream, but because everyone was really highly motivated, is becoming reality. This is a collaborative effort with partners from the African Union, from the WHO, the European Commission, where we have identified places where to build these facilities. This is the groundbreaking event in Kigali for our first BioNTech facility, which will be inaugurated later this year in December. And this is not only an opportunity to produce the next COVID-19 adapted vaccines there, but we see this as an opportunity to also produce vaccines against diseases which are neglected, vaccines which are developed based on mRNA, and therefore we also have in parallel initiated vaccine development programs for the diseases you can see here: malaria, tuberculosis, HSV2, monkeypox. We have already ongoing clinical programs, so this was also light speed, again because everyone in our company was highly motivated in all of these diseases with new antigens we have designed based on our learnings in cancer, where it really matters to find the right antigens and T-cell antigens in this regard. With this, I would like to stop and confirm that, yes, breakthroughs in science happen by bringing contributions together that seem small, and by bringing people together, stakeholders, which all are committed. So thank you for your interest.
Today, as it's a short talk, I'll focus on what we do different than BioNTech, and then we can discuss more afterwards. So what we have focused on in NICODE is to control where the antigens are taken up, and to do that, as you nicely said, the APC is a very important cell. The antigen presenting cell is the general, and what we really want is to get the antigens to be properly delivered to these antigen presenting cells, and that's what we worked on in NICODE, is to create this molecule where the antigen is directly linked to a targeting unit, which can be any molecule that binds to surface receptors on antigen presenting cells, which is the orange part here. It's connected through a dimerization unit, so that we can have 2 different targeting units that binds then to neighboring receptors on the surface of the antigen presenting cell. This molecule we encode for the programs we have in the clinic today as a plasmid DNA-based vaccine. But as you see, we can also make these vaccines as an mRNA-based vaccine, or even encode it in a viral vector, or purify it as a protein vaccine. The targeting units that we are using can be diverse, so we can use natural ligands like cytokines or chemokines or bacterial proteins as a targeting unit that will bind to surface receptors. And by that, they will bind to different subsets of antigen-presenting cells that all express different receptors. And they will be taken up with different kinetics and into different compartments. And by that means that when we choose different targeting units, we can achieve a different immune response profile towards the antigens. So in cancer, we have worked for a long period to find a targeting unit that gives us the opportunity to see as broad and strong CD8 T cell responses as possible. And we identified a chemokine called MIP-1 alpha or CCL3L1, which is an inflammatory chemokine, which we incorporated in our cancer vaccines. And when we do this, we inject the plasmid DNA intramuscularly, and then the muscle cells take up the plasmid DNA, could also be mRNA, which I'll show you soon, and then start to produce the vaccine molecules that we call the vaccibodies. And since the chemokine has these chemoattractive abilities, first of all, what happens is that we see an influx of these antigen-presenting cells coming into the injection site, meaning it will be a very short distance between the muscle cells producing the vaccine and the APC. And this will then constantly produce new proteins, and you will get loading of new antigen-presenting cells over time. It's also then obviously internalized after binding to the receptors, and we've seen through many mechanisms then that when we bind to these particular chemokine receptors, it's routed through the cross-presentation pathway very effectively, so that's the reason why we see a broader and stronger CD8 skewing of the immune responses. This is something that we then apply as a platform or a sub-platform, so all our oncology programs work with this particular targeting unit, and so far all also been working on the plasmid DNA format. And this is something we use for off-the-shelf cancer vaccines, as well as then fully individualized cancer vaccines, as also we heard earlier today. We have then partnerships with Regeneron, both in oncology and also a couple of infectious disease programs, and also with Genentech on individualized cancer vaccines. And as I'll touch upon in the end, also some interesting autoimmune programs where obviously we changed the targeting unit. So I'm not going to bore you with all the clinical data generated so far, but VB1016 is our wholly owned lead assets where we use this particular technology to target HPV-16-induced cancer. And this trial, we're using VB1016 in combination with atezolizumab in patients with recurrent metastatic cervical cancer. And we were very enthusiastic about the data generated so far. We see, compared to what we see in atezolizumab alone, and other checkpoint inhibitors alone, more or less a doubling of the most important clinical parameters, both objective responses, progression-free survival, and overall survival has not yet been reached, and estimated to be approximately 25 months compared to 10 to 13, 14 months with what we've seen in other trials. So VB1016 is a huge focus for us to move forward in different indications. When we also work with these fully individualized cancer vaccines, as touched upon earlier, we all have different mutations in all our cancer cells. So here we are then creating one vaccine per patient based on the particular mutations in each patient. This is trained on our own neoepitope prediction model. And what we've seen in the trial so far is that we generate, as we've seen also in preclinical models, very broad vaccine-induced T-cell responses, and also very long-lasting. So we can see these T-cell responses remain. Here we tested up to at least 1 year after last vaccination. So recently, we also wanted to test if we can create these vaccines also as mRNA-based vaccines. So we worked with providers here to test this particular construct. So this is with 20 neoepitopes in the vaccibody format, and then compared with a more normal standard mRNA-based vaccine with the exactly same set of neoepitopes. And we see, and this is something we've seen when we compare with the DNA versus mRNA, but when we add this targeting unit, we do see a more fast immune response to the antigens, but importantly and stronger in totality. But we also very importantly see an immune response to multiple neoepitopes that are non-immunogenic when they're not targeted to these receptors. And this is something we've seen before when we compare DNA and other formats, but now we have these data head-to-head where the only difference is the targeting unit. So it's also very interesting for us to get those data out there and explore different modalities. Recently, we've also worked on the technology in two different additional aspects. So one is to target the antigens to completely different subsets of antigen-presenting cells, tolerogenic antigen-presenting cells with different receptors. We're using all the know-how we have, so we can use natural ligands as well here in the targeting unit that will bind to surface receptors on then different subsets of antigen-presenting cells and provide a completely different signal when binding to these cells and taken up through different compartments. And in this aspect, then we can achieve regulatory T cells towards the antigen and not the CD8 dominating T cell response we see in cancer. We have also implemented a second generation where the plasmid DNA in this case can also encode additional molecules, so at least up to 3 different proteins, for instance cytokines, that will then also work on the same antigen-presenting cell as gets the antigen delivered with the Vaccibody molecule. And this regulatory T cells can then inhibit disease-specific T cells. So we have then seen both in this EAE model, which is an MS-like model, and type 1 diabetes, that we can really change the immune response. Let's see, here in the bottom, we can see it both with the Vaccibody alone, and here with the Vaccibody with these additional cytokines, where we see no sign of disease. Here, I also think it's very interesting to see what the targeting unit can do, because here in this aspect, if we increase the dose of the antigen of the vaccine, we can completely kill antigen-specific interferon gamma, while we obviously see the complete different when we do the other targeting unit for cancer. So just to sum up, when we target antigens to antigen receptor cells, we find this is a very interesting model to achieve the desired immune response to the antigen, which we can then apply across cancer, or now tolerance can obviously also be customized for infectious diseases in the future. Thank you.
So I will talk about how you then, when you do these developments, new technologies, we already seen early clinical trials data, and of course, a big impact, population health impact of the COVID-19 vaccines. But how can we think more generally about ensuring that these technologies actually work and demonstrate that across the world? And we'll talk about both vaccine trials as well as therapeutics trials in a short presentation, just to be clear. And first, to actually develop new technology in health takes a long time. And the median time for a vaccine was 8 years before the tremendous breakthroughs we saw during COVID-19. So just thinking of the different ways the COVID-19 pandemic would evolve early on in actually, the time you make your decisions regarding starting, the most realistic ambition was really this would come after the first wave. And this is sort of 1 year, and this is the potential seasonal waves coming later. Speculations at that time, because it takes time. And I will in particular now talk about the need to test whether these works. It's not only about immune response, it's about safety, but it's about how can treatments and vaccines work. And the background why Norway has been very involved and engaged in this is that we, from the Norwegian Institute of Public Health, took an active role together with the World Health Organization, MSF, Medicines Without Borders, and the Guinean health authorities in the big Guinea and big West African Ebola epidemic in 2014 to 2015, and where we managed to demonstrate that it is possible to do clinical trials in the midst of also localized emergencies in developing country settings. We used the ring vaccination approach, vaccinating around new cases of Ebola, and in a way a forest fire strategy that was used to eradicate smallpox, but here used in a controlled experiment, a controlled trial. And as you see, so of course it takes some time to generate an immune response, but then after the window we had decided, there was no cases in the vaccinated group. And I'm not going into results here, but the point here is that it took time because actually then we needed, same as for COVID-19, we needed to use vaccines that were in the fridge, had tested on monkeys, non-human primates, but never really gone into clinical trials on humans. All of that needed to happen, and what you see here are the case numbers in Sierra Leone and Guinea. And the point that it takes time to plan and conduct both phase 1 and then phase 3 trials. And the 90 rings, the ring vaccinations we did, happened just on the shoulder of the pandemic. And we were lucky, in a way, that we were in Guinea and not in Sierra Leone, in Liberia, because at that time, the pandemic had already, or the epidemic had already been controlled by public health measures. So how could you shift that window? We need to be prepared, and we need to make sure that we can test vaccines for immunogenicity and safety before an epidemic happens. To do that, you need to think differently about the normal divide in technology development between private and public. You need to actually do more on the public side. You need to still collaborate, but you need to invest more and move to actually take some more responsibility on the clinical trial element and technology development. That was really the background for creation of CEPI, and the CEO of CEPI will come after me and I'll tell more about that. But the success from the Ebola outbreak was that created a political momentum to establish an organization to actually deliver on this faster. What also happened was that the WHO created an R&D blueprint, a program really to start thinking about how can we prepare on all the outbreak-prone epidemic diseases. Ebola, yes, Lassa fever, Nipah, and so forth, but also pathogen X. And the first pathogen X was really COVID-19. I will not go into detail. I will focus briefly on therapeutics clinical trials, because a new pandemic, no specific treatments for COVID-19. We need to test what we have. And we need to repurpose established drugs and see whether they can work. The problem is that this was done all over the world in very small trials without sufficient power to really identify whether any of the drugs really worked. So more than, around 1,800 trials were registered. At least we know almost 1,000 were recruiting patients. But the problem, they couldn't really answer that question. So that's why WHO, as the leading authority on health matters, consolidated trials and actually made a world collaboration across countries. Managed to recruit more than 11,000 patients, almost 12,000, from 29 different countries across all the 6 WHO regions of the world. The data and more than 450 hospitals during the early phase. And this is the geographic distribution. This happened across the world. In addition, in the UK, as one single country, they managed through the recovery trial to actually recruit the same number. And that demonstrates also the possibility of doing that in individual countries if you are really systematic by including more or less all patients that are eligible to go into clinical trials when there is equipoise, when there is no knowledge what can work. So we can do that much better. And that's why Europe is now setting up much more of a collaborative system on how we can do large-scale clinical trials, both on vaccines as well as therapeutics in the future. These are just some of the organizations involved early on. The different trialists were forced, and I mean it, they were forced to collaborate because in the beginning that was a lot of competition. That's why we had all these small trials. And now Europe is setting up more of a concerted action by a top-down steering committee with money and funding, a coordination committee, with a pandemic preparedness partnership, a funding mechanism across member states and the European Commission to fund both in preparedness as well as during emergencies, and then with a researcher-led coordination mechanism across clinical trial networks. And this mechanism is led by the Norwegian Institute of Public Health, being a neutral actor in many ways in a small country outside EU, but still actively involved. Thank you.
I wanted to talk a little bit about, first, very briefly, to talk about our partnership with BioNTech. We have recently established what we call a strategic partnership with BioNTech. It's a strategic partnership because we recognize the potential value of an ongoing relationship beyond just a single vaccine development project. In CEPI's early years, we established contracts with private sector partners typically to develop individual vaccines. But I think all of you can intuit the value of the technology, the platform that BioNTech has developed, and the manufacturing innovations that BioNTech has developed. And we certainly recognize that. And so we've set up this important partnership, which we've only announced quite recently. That partnership, and I'll talk about this in a minute, is going to focus initially on supporting the BNT166 product, the monkeypox or mpox vaccine that BioNTech is developing, and we'd like to see that move forward. I'll come back to that in just a minute. COVID, the response to COVID, of course, was unprecedented in its speed. We were faster than we had ever been before in delivering definitive medical countermeasures like vaccines, but we were not fast enough. And Professor Türeci already mentioned the challenge with equity, and we did not, even though we distributed the vaccines globally faster than we had ever distributed life-saving medical products, we did not achieve anything like equity or fairness in the distribution of those products. And there are lots of underlying reasons to explain that. First and foremost, I think, in any situation where something that is life-saving is initially characterized by scarcity, it is likely that those with resources will use them to secure that product for themselves. And of course, we saw that, but we addressed the scarcity challenge, and we still didn't achieve equity. And there are many links in the chain of production and delivery of vaccines before you get to equity. And if any one of those links is broken, you can produce outcomes that are not satisfactory. So for next time, and there will be a next time, and all the indicators that we have suggest that the next time may be much sooner than we like. There are a number of global trends, not least climate change, but also patterns of human activity that seem to be accelerating the rate of emergence of new infectious diseases, leading to more outbreaks, outbreaks that disperse more quickly, outbreaks that are larger, and ultimately more opportunities to encounter pandemic pathogens. And COVID, of course, was the second pandemic of the 21st century already, after the 2009 H1N1 pandemic. We have to recognize that pandemics' risk is a constitutive part of the world that we have built. And we have to address that risk systematically. We have to be better prepared to respond in the future. We have to respond more rapidly and more equitably if we are going to occupy the world that we have built for ourselves. I think all of us have internalized, where cybersecurity is concerned, that if we want to live in a digitally interconnected world, we have to address the risks of cybersecurity, and we have to pay for that, and we accept that as part of living in a digitally interconnected world. We live in a biologically interconnected world, and we have to make appropriate provision for that. My organization, CEPI, coming out of the pandemic, we were very involved in the response to the pandemic, but I won't go through all of the details. We were, I believe, the first public sector organization to fund the development of vaccines. We made our first investments on January 23rd, 2020, and ultimately invested in a large portfolio of products. We also helped set up COVAX, which was an effort to offset the inequities that we anticipated. We only partially succeeded in doing that. A lot of lessons to be learned. But coming out of the pandemic, we have reflected on what preparedness should look like. And the first vaccines that were authorized by a stringent regulatory authority, of course, was the Pfizer-BioNTech vaccine, which was authorized by MHRA, the English regulator, I believe on December 8th, 2020, if I'm remembering correctly, which was 326 days after the viral sequences were released, a little less than 11 months after the viral sequences were released, which was astounding. But as I said, it wasn't fast enough. And so CEPI has set a goal, an aspiration, but we think a credible goal of reducing vaccine development time to 100 days. And by saying that that's a credible goal, we have actually thought through this concretely, how that might be achieved. And we are now working systematically to begin to make investments in the types of infrastructure and initiatives that would lead us to be able to deliver a vaccine that rapidly. We mean that goal very seriously. It is not just a slogan. It is something that we are working to achieve, and an important element to achieving that will be what we are calling the Global Vaccine Library. So what is the Global Vaccine Library? It's actually I would describe it as a logical extrapolation of an idea that was first articulated in actually as recently as 2017 by Barney Graham and Nancy Sullivan, who are scientists, many of you may know them, at the Vaccine Research Center at the National Institute for Allergy and Infectious Diseases in the US. And in 2017, they published a paper proposing an approach to preparedness that looked at the universe of threats. And they pointed out that there were about 25 families of virus that were known to be able to cause infection in humans. Only about 270 viruses have ever been documented to cause infection in human, and they are distributed over those 25 families. And they proposed that we could, by selecting what they called prototype pathogens, develop generalizable vaccine design solutions against categories of virus, and that we would use those generalizable designs in combination with rapid response platforms. mRNA was already well on the horizon. When I was in the US government, we had invested in the development of an mRNA vaccine against Zika in 2016, for example. But that we would use those generalizable solutions to map the threat space, as it were. And when a new virus emerged, we would take a vaccine that we had developed and that was from a related virus, or hopefully a closely related virus, and adapt it very, very quickly. And a core idea behind that idea that these generalizable vaccine design solutions might be possible was something deriving from work that had been done at the Vaccine Research Center by Barney and by Jason McClellan and other scientists, that started by looking at respiratory syncytial virus, very well-known virus, that has a protein on its surface which when it binds with its target cell, changes conformation fairly dramatically. And they had discovered, publishing a paper in Science in 2013, that if you inserted prolines at critical sites within the pre-fusion form of the protein, you could stabilize the pre-fusion form of the protein in a way that left it exposed to the immune system and produced highly immunogenic vaccines. And that's actually the basis of the RSV vaccines that have now been licensed. And they also realized that these surface proteins, so-called class 1 fusion proteins, were found in many families of virus, including, very, very fortunately, coronaviruses. And so at VRC, they selected coronaviruses as because they were concerned about the risk of coronaviruses as the target for proving this concept of the prototype pathogens. And they had been working with Moderna to develop a coronavirus vaccine with a stabilized pre-fusion spike protein against MERS. And when COVID emerged, literally within 36 hours of getting the sequence, the first generation Moderna vaccine was designed. And then it took almost a year to test it, but they designed that vaccine in hours after the sequences were available. And NIH, having the intellectual property to the protein key, non-exclusively licensed that to Moderna and to many other partners. Do I have, how many, couple more minutes?
Not really, but...
Okay. I'll be very quick. I'm actually almost done. So this was actually a proof of concept for the idea that you could develop these vaccine design solutions in advance and use them, turn them very quickly when you had access to appropriate rapid response technology. And our concept of the vaccine library is to extrapolate that, to engage partners. And now there are many, many institutional partners working on medical countermeasure development around the world in a deliberate effort using the viral families approach. And WHO is about to publish a viral roadmaps for the different viral families, working with partners like BioNTech to begin to accumulate these generalizable designs. And so the wonderful thing about the set of presentations that you have seen tonight is that we have talked about advances in vaccinology, advances in immunology, advances in manufacturing, and advances in clinical trial design that have enabled rapid development of vaccines. And now we need to, in a very deliberate and intentional way, to bring those together, Özlem, as you said with your picture of the tiger, bring those together to create a global capability to respond much more rapidly, much more efficiently. And as the BioNTainer facility that will be opening in Rwanda in just about a month demonstrates, much more equitably. So thank you. Sorry to go long.
But before that, I'm going to have the first questions from the floor here, and I've specifically asked Rebecca Cox, who heads the Vaccine Center, Center for Influenza Vaccine at the University of Bergen, to start. So Rebecca, please.
Well, firstly, I'd like to say thank you very much, particularly for that fantastic talk and the interesting talks we've had. So I head the Influenza Center, so for many years I've worked on pandemic preparedness. I have a sort of three-part question to you as a panel. The Gates Foundation now has stopped really work on universal influenza vaccines. Of course, influenza is a big challenge, and it wasn't on the list of pathogens that you are working towards a vaccine, because they believe messenger RNA vaccines are the answer. So my first question is, yes, it's a paradigm shift. Do you think that that is true and that we shouldn't be really working towards vaccines that provide longer and broader protection? If you think that, how are we going to make sure that we have that broader protection? Because you've talked about antibodies and T cells, and we need probably several layers. And my third part of the question is, it saddened me greatly. In 2009, there was no global equity, and we had a little bit better now. But in order to have global equity, and in order to actually have vaccine manufacturing, you need to develop markets. So after the 2009 pandemic, there were no markets, even though they rolled out influenza vaccine. So we need to think long-term about how we can make this sustainable. So is messenger RNA the answer? Do we need a sort of 2 or 3-prong delivery? And how do we ensure that it is sustainable?
I'm sure Özlem would like to answer at least parts of the question. It's a 3-prong question, and maybe others would like to sort of then fill in on some parts, please.
It may well be that influenza was not on my slide, because I just forgot to put it on the slide. Because we actually are working with our partner Pfizer on a flu vaccine and also on a flu-COVID combination, and that's definitely an important path to go. I think that especially for influenza, mRNA is a wonderful platform because we can bring in the T-cell response much more potently, including additional T-cell strengths. Because there's so much work and research on flu, we understand very nicely what the T-cell epitopes are, how the immune response works, how antigenic sin plays a role, and so on. So this is definitely an mRNA vaccine case. I'm not sure whether the other questions.
Agnete, would you want to add anything, or?
Yeah, sure. I mean, I think nobody should stop. I think we're on a mission for forever in order to make improvements. So yes, I think it's great to see that things are moving in the right direction. I think we've seen some very promising results with the COVID vaccine and learned a lot about mRNA-based vaccine and genetically encoded vaccines, but there are certainly opportunities to improve both the longevity of response, whether to what extent you should have these antibody responses or another set of antibody responses, and in combination with what kind of T-cell responses and the durability. So don't stop, continue.
And then John-Arne, you indicate that you want to answer other parts of Rebecca's question.
Yeah, so instead of answering it from a vaccinology or immunology point of view, more a political economy point of view, I will not speculate why you did not have it on the slide, but one approach is that you talked about markets, and I think the thinking in health in general is that influenza can be solved by the market, and then I'm talking about the commercial market. The problem there is that there has not been sufficient incentives to actually change the technology platform that we've had used for many years. I hope that now with the breakthrough in COVID-19, new players have come on board, and that there is an opportunity to really break that sort of monopoly in many ways of the old technology, and so that we can have new vaccines. But for influenza, then, there will be a commercial market, and we don't need to worry that much about sort of creating sufficient demand. That's different from all the other sort of smaller outbreak diseases that Seppi was mentioning. And unfortunately, also the big sort of infectious diseases, first and foremost, now having a toll in developing countries like TB and malaria. So that's why it's also very important that these companies are showing commitment and dedication to working on those. But there we need a public-private partnership so that we can actually translate the public health demand to an also viable market for companies.
If I could just quickly add, you had mentioned the Gates pulling back on the universal influenza vaccines, and I mean, a lot of work has been done trying to develop those. It has been biologically difficult, but it doesn't mean that you should stop. And I would say that Seppi's perspective, I mean, we have a large program to develop broadly protective coronavirus vaccines, and we know there is biological proof of concept in the studies that were done in Singapore, vaccinating SARS-1 survivors with the BioNTech vaccine, showing that it produced a large and broad protective response against sarbecoviruses, the subgenera of viruses. I didn't have time to come back to our work with BioNTech on the mpox vaccine, but in that case, we actually are extremely interested to know if the attribute of our existing smallpox vaccines, which is that they protect against all known orthopox viruses, can actually be replicated with an mRNA vaccine. And of course, the vaccine is designed to try to elicit that broad protection. And so I think with the kinds of advances in vaccinology that we're seeing, coupled with these antigen payload delivery systems, we should pursue broadly protective vaccines. The use case for those broadly protective vaccines may simply be to stockpile them in case we have a emergence of a new pathogen while we use mRNA to develop the highly specific vaccines against whatever that pathogen is. But we need to think that through.
And if
Thanks. Okay, go ahead. Please.
And if I may add, with technologies like mRNA, which are fast, you also have a higher fidelity situation. You can start to generate the vaccine much later and have a higher fidelity on which strains are actually the relevant ones, which at the end has a higher probability to have a better product which creates its own market. Actually with variant-adapted vaccines, we are already faster than the 100-day mission, right? Yeah. So you can predict, you can define the strains 2 months prior needing to deliver the respective strain for the respective seasonal vaccine for flu, for example.
Okay, I think there were some questions in the back next. Could you please raise your hand again? Gunnveig there, and Sigrid. Maybe we take, you take both of the questions. We can even take one more if there are more. And yes, please, here. And then we hear 3 questions and then you have a response. So Gunnveig, please. Gunnveig Grødland.
Thank you. I very much enjoyed listening to all of you, so thank you for that. But I do have a question specific for Türeci. And I was wondering about the mRNA vaccines in terms of how they actually enter the dendritic cells, because you said that there was a charge difference in the lipid particle that would not only drive them towards the lymph nodes and spleen, but also into the actual dendritic cells. And I was wondering if you could say a bit more about how that actually works.
We don't fully understand the mechanism. We know that the particles, the nanoparticles, are taken up by a mechanism which is similar, which is probably macropinocytosis, which is a uptake mechanism which is constitutional for dendritic cells or other professional APCs. We don't exactly know how the mRNA is then released into the cytoplasm. That's not so easy to investigate. So how does it escape the endosome and the liposomal particle? We're working on that. What we know is that after uptake, the mRNA ends up in the cytoplasm and is expressed there.
Thank you.
Sigrid Bratli.
Thank you so much for excellent talks. I have a question of a slightly different nature. So you had a slide where you showed different types of challenges. So knowledge challenge, supply challenge, and equity challenge. I would argue that there was a point missing there, which is the trust challenge. A significant bottleneck to uptake of vaccines is lack of trust in science and technology, and also even in governance and policy making. How can we address the trust issue going forward for vaccines specifically, but also generally for science and technology, which will increasingly be part of solving societal challenges going forward. And I think it's for all the panel. Yeah.
Thanks. And I suggest we hear the next question, which is here, row 3, and then we do a little round of responses.
Gro Tunheim, Norwegian Institute of Public Health. So I have a question regarding the cold chain. Is it possible to make the mRNA stable, temperature stable? Because DNA has the advantage of being stable at room temperature, but the mRNA, as far as I know, requires very low temperatures, and also the cold chain. So for equity also, I guess it would be better if it could be, is it chemically possible to make it more temperature stable? That's my question.
We could do even one more if there are more. No. Okay. Who would like to address Sigrid's question first?
Just on the trust challenge, I think that's particularly acute in the context of proposing to deliver vaccines even more rapidly than we did. And I think in communities in lower and middle income countries, we hear always about the importance of community engagement and enlisting trusted representatives of the community. And I think that's probably also true in our own communities.
Anybody else that would like to comment on that question?
I can comment a bit from my previous role at the Public Health Institute responsible for vaccines and vaccination. And I think there is geographic diversity on the current status of trust. Definitely, we are lucky, fairly lucky here in Norway, and the data and opinion polls indicate that, and I think our experience indicate that. But still, it's a challenge. And my reading is that engagement and actually willingness to take the debates and not trying to sort of silence in any way and distance this, but actually active public debate. Of course, the pandemic in a way allowed us to have much more of a scientific and technological debate in media. In a way, we should have more pandemics in some sense, because you never see an NRK that engaged in actually bringing new science on the news. And to be honest, I think that's what is needed. It's a continuous engagement. You do a great job, Sigrid, regarding engaging the public in new biotechnology and sciences, but not trying to silence, not trying to say that these are outliers. Social media have made it more difficult because there are clusters of communication that you really cannot see fully. So this goes both for science in general, but also specifically for vaccines and access to technology. And then I believe geodiversification, and actually the work that BioNTech is doing with the BioNTainer and what CEPI is doing in general, that it creates more of a broader ownership to science and technology. And we should also respect that when in the difficult negotiations now, really trying to correct the equity and fairness issues globally after the pandemic. They don't want more of our vaccines. They don't want donations of products. They don't want a quota of 10% or 20%. And I said they, that's the developing countries, and in particular African countries and Asian countries. They want the means of production. They want to be a part of the innovation system. They're part of the R&D system and a part of the production system. And that is also a way of creating long-term sustainable trust in science and the use of technology.
To the supply chain question, mRNA is not as unstable as people think from the very early days of approving the vaccine and supplying it. Back then, we still had to create stability data. This is how drug development works. You have to first show that your vaccine is stable for this and that time, and then you are allowed to store it at those conditions when supplying it. And the data was simply not there. Nowadays, we are at a point where the vaccine can stay for several months at 4 degrees. So, and we are getting better and better. There will be other formulations, maybe even lyophilization. There are groups which are working on that. I have no doubt about that, that also the supply topic will go away at some point.
Agnete, you look like you would like to also...
I would like to. No, I think it just briefly on the trust as well from me. I think it's super important that we as scientists dare to be out there with a clear voice, because it's so easy to be a critic and be very strongly opinionated in these discussions. And it's very easy for scientists to be very conservative and say, oh, we don't have any proof that it may be, whatever. So I think that as a community, I think we need to help each other and support each other for daring to have a clear voice in those discussions, so it's more balanced in the media. And as to the DNA, I think it also goes a bit to your question here. I think we also need as a community to be aware that we do have strong progresses in some area, but we still need to focus on things that are not just further developments of what has progressed the most rapid. So for everything here also, the mRNA has its opportunities to then be more stable and to understand more how it's delivered and improve it in multiple manners, which is important, and I think we will see a lot of improvements there. I think you're very dedicated to find improvements that we can work on in the future. But I also think, for instance, DNA that's been mentioned here several times, it has some inherent properties that mRNA do not have. And you can also work with DNA to make it more immunogenic, and we have all the benefits on the manufacturing stability and the long-lasting production of protein after DNA vaccination. So it's important that the community, and I think also Seppi, to some extent, continue to support different technologies that have different starting points, so that we take advantage of all the opportunities in our mission to find the best solution in the future.
I was wondering whether the panel, since we're discussing trust and how to sort of expand, would also like to comment on, when building an ecosystem, about competence building and employment in different regions, and sort of how to, I would think that's also engaging in communities.
I can certainly jump in, and Özlem, you might want to talk about plans in Rwanda, certainly, but CEPI is committed to geodiversification, particularly of manufacturing, but we also recognize that we need to commit to geodiversification of upstream research and R&D. And WHO has recently established a biomanufacturing training hub, Center for Excellence, actually in Korea, interestingly. And so we, working with our partners in developing country vaccine manufacturers, will be working to expand access to those training opportunities there and in other training centers. Ultimately, those trained individuals, what you would prefer not happen is that those trained individuals go and get trained and then move to a high-income country and leave their home countries behind. I think there are some very encouraging stories. In Senegal, Institut Pasteur Dakar is standing up a new manufacturing facility and having Senegalese engineers and pharmaceutical workers who are inspired by the vision returning to Senegal. And I just had the chance to meet with JJ Muyembe, who is the director of INRB in the Democratic Republic of Congo, who single-handedly built that organization up over 40 years, talking about his PhDs. 80% of his talented students that he sends to get PhDs in Western universities are now returning. So I think there is a very positive story to tell about science in parts of the world that have not had access to resources before, and we should do everything to support that.
We'll take the next question, row 3 here. And if there are other questions, please raise your hand. Okay, so I suggest we take 3 questions. You, Jens Petter Berg, and then back to Rebecca.
Okay, I'm Andrew Whitelow. My background is in pediatrics, and I have a question about global cooperation. I mean, viruses cross borders, but we live in a divided world. The Russians produced their own vaccine, the Chinese produced their own vaccine, and knowing the rivalries and the suspicion between some countries, I wonder if the panel could comment on the challenges for producing true global cooperation in this work.
And then Jens Petter Berg, 1 row back.
Yes, that was exactly the same thing I was thinking of. That is a global trust. If you please could expand what can we do to get global trust and also sharing information on viruses, on the clinical experiments and trials going on.
And then Rebecca Cox in the middle, row 2.
Thank you. Can I come back to the question of, I think it's really important that we have global diversity, but in the long term, these manufacturing capacity needs to actually be generating vaccines that can be used locally. So we need to also, whilst we have this nice suite of vaccines that are very important and thinking of pandemic preparedness, I think we need to also think long term for it to be sustainable, that they need to be producing vaccines that are cheap and can be readily used so that they can continue to manufacture, because within 10 years we started to lose that with flu, despite the fact that there had been diversification globally.
Okay, so with the panel, I'd like to start with Andrew Whitelow's question.
I can start, and maybe start on a positive tone, because we would not have seen the publication of the gene sequence of COVID-19 without global collaboration, scientific collaboration, at least not as fast. And because that was based on, like all of you as scientists do work together, a collaboration, a network. The gene sequence was in the hands of researchers in the UK and in Australia, and they nudged and the Chinese counterpart to make it public. Why am I saying this? I'm saying this because I think that kind of work just needs to continue, and that's the science diplomacy in many ways. It's about continuing scientific collaboration, also when the geopolitical situation is more difficult. We have moved from a unipolar world to that unipolar system being actually quite not robust currently politically, and then with China coming up and a very tension technologically. I believe we cannot live sustainably long term without bypassing in a way that divide. And I'm not speaking now as Minister of Foreign Affairs of Norway. I'm speaking as John-Arne Røttingen. And I think Europe is trying to find a sort of middle ground in the way of collaborating across the divide. So then, why I'm saying this, because then we need to avoid that countries in Africa need to choose in a way. Do we rely on China? Or do we rely on the sort of traditional Western countries? Fortunately, they do not need to do that currently. But a couple of days ago, Africa CDC in Addis Ababa, all their buildings, all their infrastructure, all their new lab is built by China. And they are putting a lot of investments into Africa, in particular on the infrastructure side. I think we just need to collaborate and work, and that's at least on the global scene. If I can briefly on sorry for being long, but on the point Rebecca made, it's definitely true. And actually, I think the long-term issues is always the risk-based, the health security issues, because we never know when those platforms or those pathogens will need to be used. So we need to have a short-term perspective, and that is actually making sure that we can produce vaccines that are used tomorrow and the next day, routine immunization. The problem now is that we have a lot of investments in the new platforms and technologies, which is important and we should continue. And then we have traditional vaccine platforms, and we invest in manufacturing capabilities on the new platforms, but there will be no demand and market for that tomorrow. So that's what Gavi is seeing. I'm on the board of Gavi. The board will most likely approve approximately $1 billion incentive mechanism for onshoring vaccine production in Africa. We will discuss this in Ghana in early December. And that is to really pay the premium price, the additional price that any new producer will need, because it will be more costly to come into the market and compete with Indian and Chinese manufacturers, at least as a starting point. So what we haven't cracked is then how can we use these new platforms for routine immunization. And I hope some of the vaccine or the pathogens that BioNTech and Moderna and other mRNA-based technology platforms can actually tackle what we also need every year. Yeah.
Okay, other panel members that would like to respond to this?
Just very quickly on the sustainability question as well. It is critically important, and facilities that aren't used won't be able to perform in a crisis, obviously. We actually saw that in the United States. A cold facility that they tried to stand up immediately had quality problems, and it led to vast amounts of waste. One of the, during the pandemic, Johnson & Johnson did a tech transfer of its COVID vaccine to Aspen Pharmaceuticals in South Africa, which was a biologics manufacturer, but not a vaccine manufacturer. And with an investment on both sides, including Aspen, of hundreds of millions of dollars to stand up the vaccine manufacturing, which was the first production of a COVID vaccine in Africa. And by the time they were able to produce, demand for the vaccine had evaporated. And there was a very significant risk that that infrastructure would be repurposed for commercial uses that were non-vaccine related. And so CEPI and the Gates Foundation helped to underwrite technology transfer of routine immunizations for filling and finishing in the Aspen facility from Serum Institute of India. And that will provide ongoing production at that facility. And so it is a way, we need to be very clever to think about how we achieve sustainable solutions. And one of the things I'm excited about our partnership with BioNTech is potentially working with the facility in Rwanda for purposes that are important to us and important for health security, while that portfolio of other products is being expanded.
Yes, if I may add to that, I think it's important to look from the ecosystem perspective. And the facility is an important part of it, but it's not the facility which needs to be built and protected. The question is really, how do we build an ecosystem and a functioning business model for the public-private partnership which needs to be formed with multiple stakeholders. And I think these are wonderful examples which you just described, which show that such public-private partnerships will come into situations where it's important to have a relational and not a transactional partnership, because what is happening is we are building business models together which are not there yet, which are more socially responsible than those we have. And that means it is a path which needs to be gone. There are no standard solutions there yet.
Okay, other burning questions? If not, I did promise that the panel members will have the opportunity to comment or on their presentations or ask each other questions also. So I don't know if that's. I did promise that at the start. John-Arne, please.
Yeah. So more of a comment and also to the audience, because Özlem described how the story of BioNTech started on cancer and really developed based on a strong commitment on that. We had a brief discussion earlier today, and I also know we have excellent cancer immunologists in the room and in Norway. You have dedicated yourself to understanding, I think, both the epitopes, the antigen presentation, the optimization, in a way that is much advanced compared to infectious disease immunology, at least in the standard system. And also, as you know, we measured a lot on antibodies purely on COVID-19. We need a much better and richer understanding of the immune response for infectious diseases. So with my heart on infectious diseases and global health, I think now trying to use the basic knowledge understanding from cancer immunology and continue that sort of collaboration across cancer and infectious diseases, because we heard about that inspiring startup of BioNTech that could then help us all in the world, and think how we can do that still moving forward. We need much more detailed understanding on the immune response to infectious diseases, and that may make us crack the really difficult, complex infectious diseases. So that's a call out to you and also to these excellent researchers on the panel.
And I can just corroborate that John-Arne is really serious about that, because when he hired me to be CEO of CEPI, I'm actually an oncologist.
So I think you started on closing statements here, John-Arne.
Sorry.
So, no, that's fine. I think, but I'll invite the other panel members to also then, if they don't have questions to each other, to summarize and conclude a little bit from the discussion today.
No, I do have a question.
Okay.
Or
please,
a follow-up
please,
also on your
please,
comment, and I think everyone regarding infectious diseases and the T cell component, because you also touched upon that you are in addition now adding T cell epitopes coming from additional antigens in addition to spike in the new trials, and I just wondered how your experience has been with regulatory authorities and funding bodies when it comes to the role of T cells and how to measure the role of T cells in infectious diseases for a global vaccine.
That is also a learning for regulatory authorities, but also for pharma companies which are in vaccinology, because this focus on antibodies, maybe also because it's much easier to measure them, to quantify them, and to make a regulatory path narrative out of it is there. But everyone is eager to learn. So the discussions are such that regulators in the meantime understand the role of T cells. They also understand that it's not so trivial that they can easily ask for correlates of protection, for example, because there's this polymorphism. You cannot have on a population level a clear correlate of protection, but also not the risk of population-wide evasion from immunity, which is the upside of it.
No, I think we have the exact same. We also worked on a T-cell based vaccine for COVID, and I think maybe so this is going to be difficult. I think we need to understand it's going to be difficult, and we should not ignore that we may get better protection against infectious diseases if we together as a community understand the role of T cells, and in that aspect I think also the funding institutions should be should be involved in helping us getting there and educating everyone. Because as long as we don't do the experiments and try to find any levels of correlates of protection and the role, we will not get better vaccines that also include T-cell, the T-cell side of it.
I would just to build on that, the importance of addressing these regulatory questions or regulatory science questions, those are critical to advancing products. And often funding agencies are much more interested in the discovery work or the upstream work. CEPI, because of where we sit, which is in product development, we recognize the criticality of those questions, and we do work with regulators to try to understand what are the regulatory science gaps that would enable you to make assessments more confidently, more rapidly, and we are committed to making targeted investments.
Okay, are there other people I would like to make a closing statement, or I think John-Arne, you did one.
This is really difficult because we have covered so many things very comprehensively and had a couple of questions which really touched the very relevant aspects. I think the only thing I can say as a closing remark is that this was a very successful meeting and panel discussion. Thank you to everyone for this.
Thank you so much. I think that sort of ends the meeting.